Hello and welcome to the first lecture of the bioinformatics algorithm class. When I started teaching bioinformatics 2 decades ago, one of the biggest challenges was to connect with a very diverse audience. I had to talk to computer scientists and biologists, to physicists and biochemists, to mathematicians and statisticians. The challenge was how to keep computer scientists focused while explaining sometimes convoluted biological concepts. And likewise, how to keep biologists awake while explaining sometimes complex algorithmic idea. I hope that in this class we have an equally diverse audience, and that's why I decided to start from a very simple problem. Here's the first problem in this class. Given a string called text1, copy it to a string called text2. And I see that some of you are already thinking about quitting this class because it's so trivial. Please wait. You might think that it's trivial, but billions of cells replicated right now in your body, your own genome, don't think so. In fact, they're trying to accomplish incredibly complex task of replicating genome. Just think about this. You need to unwrite DNA, 2 strands of DNA, and separate them. You need to make sure that there is enough free-floating nucleotides so replication can complete. Because if you start replication and you have to stop in the middle because there is no energy left, you are dead. So in this course, we will learn about this and many other challenges related to replications. In fact, replication in human genome is so complex that biologists still do not fully understand how it's done. That's why we will focus on replication of bacterial genomes that are 1,000 times smaller. The genome is 1,000 times smaller, and that's why replicating bacterial genome is a little bit easier. I'm not saying that it is trivial. And in fact, right now, there are billions of bacterial cells trying to replicate bacterial genomes inside you, because the number of bacterial cells inside you is 10 times larger than the number of your own human cells. Now, since bacteria usually have circular chromosomes, I have to slightly change my problem. Our new problem is, given a circular genome called text1, copy it to a circular text2. It's equally trivial, but it makes some slight changes in the setup that we need to address. A circular genome doesn't have a start and an end. And that's why roughly 5 million nucleotides E. coli genome has a choice on where to start replication. In this class, we will try to figure out where does it start, because all E. coli cells on Earth start replication in the same region called origin of replication. So our first problem, where in a genome does it all begin? And the only information that we have is the genome itself. Okay? So let's go ahead and let's first try to answer the question of what are the hidden messages in the replication origin that help E. coli genome to figure out where to start. And here's the first, or should I say the third already, problem in this course, finding origin of replication. I'll give you a genome. You have to give me the location of origin of replication in this genome. Does the problem make sense? Yes? Yes? You may be right or wrong, depending on who you are. Are you a computer scientist or biologist? Computer scientist. Computer scientist, then you are wrong. If you were a biologist, then you could think about an experiment. For example, you can start cutting short pieces from DNA. And when you cut a piece and the cell suddenly lost ability to replicate, it means that probably you cut out the origin of replication. But if you are a computer scientist, you should just refuse solving this problem. It is an ill-defined computational problem. So how can we turn this ill-defined computational problem into something that makes sense.